In March 1989, the Exxon Valdez ran aground, releasing 11 million gallons of crude oil into the subarctic waters of Prince William Sound. Over the first 56 days, the North Slope crude spread over 750 kilometers, driven by a combination of storm winds and currents that contaminated the Kenai Peninsula and the Kodiak Archipelago. To measure the damage, researchers relied on the standard 1980s protocol, 4-day laboratory tests. These assays measured acute exposure, focusing almost entirely on surface narcosis and immediate short-term mortality. Under this model, scientists assumed that once the visible surface slick was dispersed or mechanically removed, the ecosystem's recovery would be rapid and complete. However, years after the surface was cleared, populations in the Sound continued to crash, indicating that standard laboratory measurements had missed the most significant biological impacts of the spill. Responders initially fought the slick with traditional tools, mechanical skimmers, booms, and chemical dispersants designed to break the oil into smaller droplets. Alongside these methods, they deployed bioremediation, a biological technique using microorganisms to degrade the oil's hydrocarbons. This process, called biostimulation, involved applying nitrogen and phosphorus fertilizers to contaminated beaches to feed indigenous oil-degrading bacteria. These bacteria use specialized enzymes to metabolize simple, straight-chain hydrocarbons through a biochemical process called beta oxidation. Bioremediation successfully removed the visible light oils from the shorelines, but the heavier, more complex chemical compounds remained embedded in the environment. While microbes are efficient at eating light oil, they are unable to process the heavier, more complex molecules found in North Slope crude. These are polycyclic aromatic hydrocarbons, or PAHs. Their stable 3 to 5 ringed structure blocks microbial enzymes, resisting biological degradation. Unbroken down, 55,000 kilograms of toxic oil sank into subsurface sediment. There, large boulders and cobbles created geomorphological armoring, physically shielding the oil beneath the surface. In this subsurface layer, temperatures drop to 4 degrees Celsius and oxygen levels are low, which stalls microbial metabolism and stops natural biodegradation. This armoring also protected the reservoirs from the mechanical energy of waves and the degrading effects of sunlight. The combination of chemical resistance and physical sequestration transformed a temporary spill into a permanent subsurface toxic reservoir. This subsurface oil was positioned precisely where it impacts a specific group of wildlife, benthic foragers. Sea otters and harlequin ducks hunt clams by digging into this contaminated sediment, causing repeated contact with sequestered PAHs. We traced this exposure using CYP1A, a liver enzyme biomarker. High CYP1A levels proved these animals continually ingested petroleum nearly a decade after the spill. In contrast, species that feed purely on fish in the open water column, like river otters and pigeon guillemot chicks, showed no such exposure. This chronic exposure did not cause the immediate mass die-offs associated with the surface slick. Instead, it took a physiological toll over years, resulting in stunted growth, altered behavior, and severely suppressed reproduction across multiple species. Laboratory studies on pink salmon embryos showed exposure to weathered PAHs at just parts per billion levels reduced their open ocean survival by 50%. Long-term monitoring of the Sound revealed the spill's legacy was an invisible, multi-generational erosion of a population's ability to survive and reproduce. The population crashes also exposed a major flaw in the old risk models, the idea that species exist in a vacuum, independent of their environment. In reality, ecosystems rely on interaction cascades, where the loss of one key species can trigger a structural collapse of the entire food web. This Pacific Northwest interaction web shows how a single shore event triggers biological instability. The spill initially killed fucus rockweed, a habitat provider, and grazing limpets. Without grazers, the system experienced a massive, unstable bloom of opportunistic green algae and barnacles, filling the void. When the fucus rockweed finally attempted to return, it settled on these weak barnacles. During winter storms, the barnacles were easily ripped away, taking the new rockweed with them and restarting the cycle of collapse. Similar cascades occur in the subtidal kelp forests, where the loss of sea otters allows sea urchins to overgraze the kelp, destroying the habitat for countless other species. Oil spills delay recovery, not just by poisoning individuals, but by shattering the structural relationships that hold a biological community together. 14 years of research following the Exxon Valdez forced a complete rewrite of how we assess environmental risk. This new model moved past the assumption of rapid degradation, recognizing that subsurface sediments can protect toxic oil for decades. It also shifted the focus from short-term narcosis to the more significant long-term impacts of sublethal exposure on fish embryos and population survival. Today, this ecosystem-based framework is required to evaluate the true scope of modern environmental disasters. During the Deepwater Horizon spill in the Gulf of Mexico, these principles were critical to understanding how oil would behave in deepwater environments. Even with advanced bioremediation and dispersant use, recovery remains strictly limited by the physical and chemical parameters of the deep ocean. Understanding the true cost of an oil spill requires looking beyond the surface slick, recognizing the deep, systemic, and enduring memory of the ecosystem.